what I'm going to be talking now is more about getting a little bit into the field of artificial cells. Okay, so why do we study, or why are we interested in artificial cells? So basically, we're going to talk about the cellular gadgets, meaning what are the things that cells use, that we can use from normal cells to assemble our own cells. That's one thing. Or like, what can we learn from what the cell has, for example, membranes and proteins, etc., to also modify them and make them different. So we're getting more into the realm of creativity here, of thinking about what gadgets can we use to make our own synthetic cells. So if you go to any kind of synthetic biology talk, I'm pretty sure that Richard Feynman will come up at some point. He has this very famous phrase that says, "What I cannot create, I do not understand." He didn't, of course, set it in the context of synthetic biology, but more about, you really understand something when you tear it apart, when you try to, um, you know, you try to kind of break it in pieces and try to really, really deeply understand it. So that's kind of one of the things that are important in synthetic biology. And synthetic biology in general, I mean, as you guys have seen, it has advanced tremendously, just simply because we have technologies, right? So, for example, nowadays, in the previous
to
era,
further improve
sorry.
the throughput and preciseness.
So in the previous era of synthetic biology, we'll have to, or not synthetic biology, genetic engineering or genetic modification, people had to use plasmids and clone restriction enzymes. Right now, we can just have machines that make DNA fragments. So for example, this kind of the GenScript kind of technology that uses this high throughput way of basically just inserting the right nucleotide one after the other and making fragments, right? So there are companies where you can simply just say, I want this gene, and then they can send it to you in a couple of days. And so this has facilitated enormously, the research in this area. So this has been kind of one of the main motivations for what people are kind of so much getting into this field. And this is just kind of another video that I find interesting. This was from the Maluk Lab in Penn State. You sometimes don't have to do an actual cell. You can use inanimated particles, like in this case, there are particles, gold particles, where they use ultrasound to align them, and then magnetic fields to kind of rotate them. So basically what you're seeing there is just some rods, like, and what you see that is rotating, these are HeLa cells, these are cancer cells. So what they add these little gold particles, and start spinning the cells, right? So it's very fascinating, the kind of things that we can do these days. So this is kind of just to give you a motivation a little bit about the synthetic biology field. Natalia and Miguel will be talking about more organisms and modifying genetically, and I will talk a little bit about this too, but we're going to focus more on that. What I'm going to give you now and in the following lectures is basically, we already saw proteins, we have seen the membranes. Now let's try to understand them a little bit more physically. Can we assemble things from scratch, right? Like this example that I told you, this bacteria that had no DNA, for example. What if we can just put things inside of that bacteria and then just make it grow? Put another DNA, for example. That's what Craig Venter did, right? So I don't know, you guys know Craig Venter. So what he did basically was just took the DNA of a bacteria and then transplanted a new one, right? Artificially made. So it's getting very fascinating in this field. So as you guys already reply in the poll, like there's simple things that a cell needs. I'm calling them, I'm a Batman fan, so I'm gonna make an analogy here about gadgets. So Batman has a lot of different gadgets for different things. So as we discussed in the previous lecture, we need a suit or a membrane or a boundary. We need a machinery that copies DNA, that makes transcription, makes proteins. But we also need something that communicates with external, with the environment. The cell needs to sense things. So, and these are kind of what we're going to be talking about. But before we move to that, it's important that we have clear what are artificial cells useful for. What do you guys think? I saw a little bit on your polls, and in the poll, like you guys were saying what what you like to learn about, and I saw that some of you wanted to learn a little bit about how to make artificial cells and so on. What do you think are the artificial cells are useful? Can you write in chat or speak up?
Well,
Just
I
whatever.
think,
Yeah.
oh, sorry, what I've seen the most is in terms of remediation. Maybe you can engineer some bacterial cell to degrade a certain plastic, maybe, or another contaminant. Mm-hmm.
Exactly. So yeah, exactly. Very good. So I think there are three different kind of goals of, it says replacing cancer cells or damaged cells. Yes, correct. Or more like you can say also killing them. I'm going to show you a very cool experiment tomorrow, a bunch of cool experiments tomorrow about this. All right, so I'm just going to give you like three basic reasons. Why do we study artificial cells? So the first one is to study the origin of life. We said before, what is life, right? So it's very hard to say, but the question is, if we make, for example, membranes that move, that divide, that have a metabolism, so at what point do they become autonomous like our cells? At what point, what do we especially need to be able to make an artificial cell to actually behave like a regular cell? And that's what we're going to be talking today a little bit. But it will also help us understand how the very first cells were constructed. So I show you the work from the Jack Soster lab with his Nobel Prize winner, and they're trying to investigate that more. You can use artificial cells to kind of understand biological processes. So in the cell, as we discussed, the cell is crowded. There's a lot of different things happening. So one of the things that can help us by building an artificial cell is we can isolate different processes. Like if we want to understand, for example, transcription, then we can just isolate it completely and just understand the proteins that work in transcription, right? Because the cell has different regulation, different kind of mechanisms. But the one that is more interesting, probably for all of you, based on what you guys wrote in the polls, is that they can do things that normal cells cannot do, right? One way, as we discussed before, I show you, we can make bacteria or human cells to do things, but sometimes there's a limit. They cannot do certain things because they get sick, for example. Like you cannot put maybe a human cell to clean up the environment because they are exposed to toxic things that will kill them, basically, For that case, it would be better to just build some artificial cell, like a robot, basically, right? That takes care of that. So that's the main goal of using artificial cells. Things expand the abilities of biological organisms. So here is just my approach. I'm currently a postdoc. I'm gonna be finishing soon, and I wanna open. Sorry, there's a question. Hirsch, sure, go ahead.
Yeah. So, are artificial cells also are they like somewhat resistant to aging, or do they also age the same as normal cells, depending on how we make them?
Exactly. That's well, I think you actually answered a little bit, because it depending on how you make them, you can make them resistant to aging, right? Because as long as they can have a source of components that they can replicate, they eventually can just expand indefinitely, right? So cancer cells, for example, don't age, right? So they just can keep going and going and going. So
okay, thank you.
in principle, we can do that, yeah. So what I was going to say is that my research is mostly about, as I mentioned before, is how do the cells, if one of the main problems that we have with artificial cells is that we cannot make them divide and have DNA at the same time and expand, so that's a very problematic mechanism for making artificial cells. And I will show you experiments on this from other people as well. So one of my goals is try to understand this in a simple organism like bacteria, so that one day we can make autonomous cells that can just simply go and divide and my idea is that we can learn from the bacteria or simple organisms so that we can copy those mechanisms, make them better, and make artificial cells that are autonomous. So one of the things that I investigate is this protein, which is kind of like similar to an actin protein, that it just goes around the cell and then makes the cell wall, makes the bacteria form like a rod. And I'm not studying specifically that bacteria, but I'm trying to see how this mechanism of cell growth is basically connected with DNA replication. So this is a movie I already showed you about how the cells replicate the genome. So the idea is how these processes are connected. As you're making genome replication, the cell needs to expand and make volumes. So how do these two things are connected? So if we can find a mechanism, the genes that control that process, then we can make artificial cells that do the same thing. So that's kind of my goal. So I just wanted to show you this example, and then I'm developing a lot of synthetic biology tools. In this case, I'm kind of have CRISPR activated here in bacteria, and I develop a tool that I just get rid of it in a very fast way. So here the cells are growing, and I just flow a drug, and I just destroy it. So the idea is to develop this kind of tool so that allows you to kind of control very accurately processes in the cell. So let's talk about the gadgets. So we're going to talk first about the suit. This is, I recommend you this app. It's very funny. So this is me as Batman looking at the suit. It's just artificial intelligence replace the face on the picture of Ben Affleck. So we're talking about the membrane. So the membrane is very important, as we discussed. We have kind of one of the things that we have seen. But you can make different kinds of membranes. So there's membrane that usually cells have are phospholipids, as Miguel also was describing in the first lecture. But you can make membranes based on proteins, for example, or membranes that are mixed, a little bit of lipids, a little bit of proteins, or a little bit of other polymers, that it doesn't have to be proteins. The reason why cells use phospholipids, for example, is because it's very flexible. It can be tuned, meaning that you can increase the rigidity of the membrane. But if you look at, but they are not very mechanically stable, so they cannot resist too much pressure. That's why bacteria, for example, they have cell walls. So they have something rigid outside to protect them, right? So membranes on its own are very fragile. In our, for example, tissues, we have, for example, like in the bones, we have calcium deposits that kind of protects the cells on the bones, right, and makes them extremely strong. So the cells have come up with strategies to compensate for having a membrane that is so fragile, so weak. Polypeptides or proteins that they can have a little bit of strength in terms of the membrane. They're a little bit harder to produce, right, because you still need to have to produce a protein, and they have to assemble in the right way, right? And you guys remember with the drop, right? With this lipid drop, the drops automatically form themselves. That's the cool thing about phospholipid membranes, that they can form themselves without much help. But a protein membrane might need to have some kind of help. So you remember I show you, for example, the actin, the tubulins, that they assemble each other. So the idea of making a protein membrane would be to have a protein that assembles on its own. And this is very hard. This is when people have been working on designing kind of, people call it the protein design, meaning that you can change proteins and make protein engineering to design proteins that are not present in live organisms, that have better properties. And the block copolymers, you know, they have other molecules that are not proteins to make them highly resistant. But this is, for example, when I use this kind of membranes in humans, it's a bit harder because they you know, the body might recognize it as toxic and react. So it's not useful, for example, if you want to put it into a human being for a drug, for example. So how can we make membranes? So I show you this simple experiment with the pipette, but you know, this is not, you want to make artificial cells, we want to make a lot of them and very fast. So one thing I wanted to tell you guys, so I'm going to share with you these presentations. Please have a look at the labs that I'm showing you, these experiments. If you see something cool, check out those labs, because maybe there's something interesting that you can do internships later in the future. So that's why I put the lab names and everything, so that you can investigate more and see if you find something interesting. So this lab, for example, has this microfluidics. You guys remember that someone in the opening ceremony said that they're going to work on microfluidics. Microfluidics are very nice technologies for automating everything. So meaning microfluidics is just imagine having kind of a system where you have little channels like labyrinths, where you can pump different liquids and control it with a computer. So it's kind of a way to control the environment. So in this case, this microfluidic just makes little drops of lipids. So it's creating these membranes just like this, you know, it pumps the oil, and it has kind of a more aqueous environment where the cells in this case, the membranes cell assemble really quickly. So you can make thousands of them in a very fast way. So that's kind of an interesting way. Now in this same lab, they started to do some cool experiments. So they put actin, as you guys remember, actin is the part of the cytoskeleton. So they put the actin proteins inside of these vesicles. And as you can see them, they just move in the vesicle, right? And this is actually really cool because, as we were discussing, some of the proteins can just act without having to have the cell itself to regulate it, right? So in this case, the protein, the actin forms filaments, and they just bundle and move around. I'm going to show you now an experiment from a lab in Germany, in Munich, that I think is fascinating. So this is, I call it the Amoeba. So basically they make a membrane and they put tubulins. So the tubulins are with some molecular motors. Miguel showed you this video where the molecular motor is walking, right? So it has something like that. So, and what the molecular motors are doing is just pushing around the membrane of the cell. In this case, I mean, I call it the cell, but it's a lipid membrane, right? And I find it just very cool because, as you can see, it has movement, right? And this is not a cell, it's just simply just a membrane. You put tubulin and then with the motors, and it starts moving around. So in principle, you could make things like this in the lab that are not cells at all, that are not alive, but they have some properties from the cell. So what about communication? So this is important. As we discussed, if we want to make cells that are artificial cells that are important, for example, to deliver drugs or to get rid of something in the environment, they need to sense first if there is something, right? So for example, if you have a toxic compound in an environment and you want to get rid of it, you first have to recognize it, right? So this is kind of the key point of having a receptor or channels that allow you to communicate with the memory. So there's another cool experiment in this paper. I definitely recommend you. So I'm going to explain you what they did. So what they did kind of a photosynthetic or artificial mechanism. So they put some proteins that are sensitive to light, like rhodopsin from bacteria, and photosystem 2 from plant cells. And then basically these receptors, these proteins in the membrane, will get activated on light. And the activation with light allows to open these channels and exchange protons So, you know, in the cells, one of the things that the cells use a lot is proton flux to generate energy to produce ATP, right? So in this case, that's what they did. So when the protons are flowing across the membrane, it generates a potential that that allows this protein that I showed you at the beginning, this gigantic protein, the ATP synthase, to produce ATP. As you know, ATP is the molecule that gives energy to the reactions in the cell. So they did this just with the proteins, and this mechanism allows you to make ATP without anything, just with light, right? Of course, you have to have water, etc., but just with light and you know, ADP, of course, this mechanism can just make energy, and they can tune it with different wavelengths. So, for example, red light, it allows you to activate the proton flux so that you get ATP. If you do green light, then you get rid of the proton flux, and then the ATP is not produced. And the ATP that they use was important so that the actin, so the microtubules, can move. So I'm going to show you the final experiment. So this is a movie from them. So if you add red light, then the actin moves. If you add green light, the actin stops. If you add red light, then it starts to expand. So I will show you again. So the green thing is that you see around, those are the photosynthetic organelles. So it's really cool that only just with light, this membrane can produce ATP and control the movement of actin. So then they did this another experiment where they control the flux of ions. So as we said, this artificial cell needs to have also ions to function in some cases, to create the right potential in the membrane, etc. So in this case, for example, this is the membrane, and the actin in the cell, the membrane breaks and the actin compresses. But if you add magnesium, then the actin kind of assembles and the membrane is intact. At some point it breaks, of course. But what we're trying to see here is that you need ions for this assembly. So they made an artificial cell that can import magnesium ions in this case. So what about the machinery? So we need the machinery, right? So the machinery is very important because we need information to tell the artificial cell something to do, right? So this is really cool experiment from this lab where they try for the first time to sort of make an artificial cell that can duplicate the DNA. So you guys can see the paper in more detail, but the whole idea was to put the polymerases in a DNA template so that it can produce the DNA inside. So if you see here, the bubbles are empty, and the green dots is the DNA that's being produced. So if you add nucleotides, then you see production of DNA. But if you don't have nucleotides, then the membrane is just empty, right? So that's kind of an interesting. It's not the perfect experiment, I think, but it shows us that it's possible to sort of make some DNA in there. Now the question is, once it makes the DNA, then the membrane has to expand because the DNA is taking space, right? The DNA, as we saw, or RNA in general, they're big. They're way bigger than proteins, so they can quickly become very spatially, problematic because it occupies too much space. In this experiment, they just synthesize proteins. So here they synthesize YFP, it's a fluorescent protein, and the liposomes they have ribosomes inside, and they make the protein inside of a vesicle without anything else, like no other organelles or anything. Now, what about cell division? I show you guys about cell division. these people, I recommend you also. This lab has really cool experiments and interesting questions in Netherlands, in Delft. So they're trying to do kind of what happens if you put, I told you about this polymer in bacteria that makes constriction, that makes the cells divide. So they put this polymer inside of the liposome, right? And they put some kind of adapter, which is FTSA, doesn't matter the name. And the idea is, can we make a liposome divide? And now the question is, what do you guys think? Do you think it's possible to make a vesicle divide or not? You can say in the chat or speak up. Are you guys falling asleep?
Well, if you're able to direct the polymerization of some microtube, some components of the cytoskeleton, I guess you might be able to guide cell division in an artificial cell or a liposome.
Right. In principle, you will think that, but actually nobody has been able to. So, but what this lab managed to do is they put the FTSC, which is in purple, and you will see that it will make like a little vesicle there. You see? It's like a neck that is pulling, and then the membrane is like expands. But they haven't been able to make something that just cuts the liposome in half. It's very hard actually, because this protein, because imagine like if you have, for example, a balloon and you squeeze it, right? You make it squeeze, 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 but you need a pulling force to make it break, right? And we don't have that. So that's actually, it looks very simple, but it has been extremely hard to make. And that's why it's very hard to make artificial cells that are able to divide. So, and just to finalize this lecture, basically, what are the current limitations of artificial cells? Coupling growth, division, DNA replication is challenging. I show you a little bit of what is possible. Environmental sensing is also very difficult too, but there are some progress. Production of more complex protein machineries inside of the liposome is what it's important for the future. And this is still in progress because we need to handle that. It's going to get crowded. So we need to be able to, the cell does this very easily by, for example, having organelles or, pushing things in one side and just make everything work in a specific part of the cell so that it doesn't perturb anything else. So the cell does this in a very smart way, but we need to figure out a way to do this in artificial cells. And also we need to be able to make artificial synthetic cells in a very, very high throughput way. Like we need to produce them a lot. So one example of this is the Pfizer or Moderna vaccines for coronavirus, right? They are basically, we'll work with this in the other lecture. But this has been problem challenging because you need liposomes, you need lipids, and actually they're having problems because the lipids that they use to make the vaccine are they're not, so they're becoming scarce, so that we don't have enough of them. So I mean, luckily it's not gonna be a problem, but they're seeing some problems. So we need to be able to make this in a way that it's, can you produce a lot and it doesn't create problems.